So hi everyone, thank you very much for the opportunity to record this presentation online. So the topic is audio frequency discrimination in an oddball paradigm, an ERP study. So this study is focused on the P300 event-related potential component, which has a well-established role in literature, and it tends to peak about 300 milliseconds after stimulus onset. The P300 is considered to be an endogenous component, which means that it has more to do with how the brain responds to stimuli rather than their physical characteristics. In this study, we use the oddball paradigm, which has been also commonly used to elicit the P300 component. And in this paradigm, usually, there's a sequence of stimuli, which can be auditory, visual, or somatosensory, that participants observe. And interspersed, there are slightly different or very different stimuli, so-called deviants. There have been several theories which try to explain the underlying mechanisms which the P300 reflects. One of them is called the context updating theory, according to which the brain builds models about the world, and it continuously generates expectations about what kind of things it's going to encounter in the near future. So in this case, if the expectations are violated, the brain has to update them, and the P300 reflects this updating of expectations. Another theory is categorization, which essentially says that P300 reflects processes involved in categorizing stimuli, such as perception, attention, and working memory. In this study, we used the an auditory oddball paradigm with three different types of stimuli. The first one was standard. These were auditory beeps of 1000 Hz, and the easy deviants and the hard deviants. And the reason why we call these easy and hard is because we kind of expect in advance that participants will find it easier to discriminate between the easy deviants and the standards compared to the hard and the standards, simply because the hard and the standard are really similar to each other. There is just a 30 Hz difference between the two. So naturally, it should be harder to discriminate between these two. In addition to EEG data, we also took the reaction times, and we predict that it will be more difficult and people will be less certain about the hard deviants, so the reaction times will be slower compared to the easy deviants. And in terms of these theories, we predict that since the hard deviants are very close to the standard deviants, then the brain doesn't really need to update its expectations so much, so the mean amplitude of the ERP response should be smaller for the hard deviant compared to the easy deviant. And with the categorization, within the categorization framework, we would predict that the hard deviants elicit a smaller match between the recognition networks in the brain and the actually auditory tone. So as with the context updating theory, the mean amplitude is predicted to be smaller for the hard deviant. So we recruited 16 participants for this year and 12 from previous years. And there was an issue with accuracy, because in the previous years there were 4 participants which had accuracy at detecting the hard deviants very near 0. So I really wonder why the experimenters did not check whether the participants could actually hear the hard deviants and continued with the analysis, because the data are obviously useless. So we used the oddball paradigm. There were 1000 trials in total, 75% of which were the standards, and 12.5 hard, 12.5 easy, to make sure that there were no effects of different probability on the ERP response. The procedure, that is pretty clear. In total, participants spent about 20 minutes in the lab, but of course they could take breaks at their own discretion in between the blocks. This year we recorded 32 channels. Previous years had 64 channels. In addition to the SCARP electrodes, we also used the mastoids and the VEOG and HEOG to look at the vertical and horizontal eye movements. Data analysis was obviously done in the EG and ERP lab. I personally reduced the channels to about 16, focusing on the central and parietal sites. And from the standard pre-processing steps, I would like to mention mainly filtering, because I started with a high-pass filter of 0.1 hertz, but the data turned out to be incredibly noisy, so I applied also a low-pass filter of 30 hertz. And when I did the artifact rejection, some participants, 4 participants, had exceeded 30% of artifact rejection rate. And when I visually inspected the data, I saw that the reason was mainly that some participants just blinked in just about every single epoch. They pressed the mouse button and then they blinked. Some people seem to have a talent for this. So the natural option was to run the ICA, independent component analysis, and I managed to reduce the average total artifact rejection rate to about 2.6%, which I consider a success. So no participants had to be excluded in the end, and except for those who had low accuracy, and 11 participants remained for the analyses. And if for the analyses, I firstly visualized the ERP data. Here you can see the waveforms for the CZ electrode, where there is a clear difference between the easy targets, which is the red line amplitude, and the hard targets amplitude, that's the black line. And this is kind of confirmed by the SCARP distribution, where you can see greater positivity elicited by the easy targets over the parietal and central areas. As for the reaction times, then these confirmed the initial hypothesis. So indeed, participants reacted slower to the hard deviants compared to the easy deviants, indicating that they were probably less certain, or it took more time to process the hard deviants. Then I looked at the statistical ERP results, and to help me choose the appropriate time window for the analysis, I ran the mass univariate analysis, and I ended up with a time window of 250 and 400 milliseconds. I found I ran 2 times 12 repeated measures ANOVA with condition and electrode as factors, and the condition was found significant, which means that indeed the amplitude for the easy deviant was larger than the mean amplitude for the hard deviant. Electrode was also significant, indicating that there was certain SCARP distribution. Not all electrodes showed the same data, and there was also an interaction between the condition and electrode, which implies that the SCARP distribution changed with the condition. And you can take a look at this graph below, which basically sorts the electrodes from the front areas all the way towards the occipital areas. You can see there's almost a linear trend where the mean amplitude for both conditions increases as you go from the frontal to the occipital, but not except for the occipital electrodes, which is the last one here. The farther you go backwards, the greater the amplitude, approximately. So to wrap this up, because I've run out of time, I think, well, the data supported the initial predictions, so there seems to be some kind of updating of mental representations in the brain, which is more prominent for the easy deviants compared to the hard deviants. The major limitations of this study were the accuracy and the artifacts. It would really be great next time to not to record useless data. The future research, in my opinion, could take a look at the relationship between the mean amplitude and the audio frequency, because in this study we had just two different kinds of deviants. But what would be interesting to see is if there is, what kind of functional relationship there is between the numerical frequency and the mean amplitude of the ERP response. And another thing this study hasn't really helped us to understand is how to distinguish between, hasn't allowed us to do, is distinguish between competing theories about which brain processes underlie discrimination between the two stimuli. And yeah, on this note, I've run out of time. Thank you for your attention, and if you have any questions, then please email me. I'll be very happy to answer. So, thank you.